Welcome to the Explainer. Today, we're going to tear apart a real GCSE biology exam question. We're not just looking for the right answers. No, we're going to get inside the examiner's head, figure out exactly how the mark scheme works, and learn how to snag every single point available. So here's the game plan for today. We're zeroing in on question 05 from a recent AQA paper. We'll start with the basic biology, then dive into the experiment and its data, and we'll wrap up by figuring out what to do next. It covers like all the key skills. Let's get right into it.
Right, so this entire question, from start to finish, is all about one very important enzyme, amylase. We'll kick things off at the fundamental biology before we see how it actually performs in a lab setting.
All right, part one. Think of these first few questions as the warm-up. They're testing the stuff you should already have in your back pocket before you even glance at any charts or data. Let's see what they're asking.
Okay, first step, a classic one-mark question. It's basically a quick knowledge check. They tell you amylase is a polymer and ask what it's made of. The trick is to connect the dots. Amylase is an enzyme. Enzymes are proteins. And what are proteins made of? You got it. The mark scheme is looking for one specific answer, amino acid or amino acids.
Now this one is interesting, not just for the biology, but for how they mark it. So, to get the full 2 marks, you've got to name all 3 of these: the salivary gland, the pancreas, and the small intestine. But hey, here's a little bonus. If you can only remember 2, you still walk away with 1 mark. Not bad.
All right, now for a bigger 3-mark explanation. They want you to break down the lock and key theory. It's a step-by-step process. First mark, just for saying the starch, which is the substrate, latches onto the enzyme's active site. Simple enough. For the second mark, you have to explain why it fits, because their shapes are complementary. It's like the perfect handshake. And that third and final mark comes from describing the result. The enzyme does its job, a chemical reaction happens, and the starch gets broken down into smaller sugar molecules.
Okay, let's keep it moving. Now we're shifting gears from pure theory to a practical application. We're going to see how all that knowledge gets put to the test in a real lab experiment.
So, this is the exact setup they show you in the exam paper. Pretty standard stuff, right? You've got your test tubes with starch and amylase, all sitting nice and cozy in a water bath. The whole point of this investigation? To see how changing the temperature affects how quickly amylase can break down starch. You know the drill with any experiment. If it's not a fair test, the results are useless. And that means you have to control your variables. The question asks for 2 things you'd have to keep the same. And look, the mark scheme gives you a whole menu of options to choose from. For example, you absolutely have to use the same volume of amylase and starch every single time. Why? Because otherwise, you wouldn't know if it was the temperature or just the amount of stuff you used that changed the result. Just pick any 2 from this list and you've got the marks in the bag. This is a classic question. It's all about testing if you understand the process of a good experiment, the little details that matter. So why the 5-minute wait before you mix everything together? The mark scheme gives a really precise answer for this. It's all about letting the solutions get to the same temperature as the water bath. You want to make sure the reaction kicks off at the exact temperature you're trying to test, not a mix of room temperature and whatever the water is.
And now for the juicy part, the results. This is where you switch from just spitting out facts you've memorized to actually acting like a scientist. You've got to look at the data from this specific experiment and figure out what it's telling you.
Okay, here it is. Table 3, straight from the exam paper. It shows how long it took for all the starch to disappear at different temperatures. And you know the starch is gone when the iodine solution stays that yellow-brown color. Now look closely. See what happened at 5 degrees and 80 degrees? The reaction just never finished. That's a huge clue. So, question 5.6 asks you to basically summarize what the table shows. It's a 1-mark conclusion. And if you want the perfect answer, the one that ticks all the boxes for the examiner, you say this: the enzyme's activity goes up as the temperature increases. It hits its peak, the optimum, at 35 degrees, and then it drops off.
And here we go, the big one, the 5-marker. This is where you can really rack up the points. They want you to explain what was going on at those 2 extreme temperatures, 5 degrees and 80 degrees, where, remember, the reaction didn't finish. To nail this, you've got to pull a few different ideas together.
So, how do you tackle a question like this? The best way is to break it down. Deal with each temperature on its own. At 5 degrees, it's all about energy, or, well, a lack of it. The molecules are moving so slowly they just don't have much kinetic energy, so they're not bumping into each other enough.
But at 80 degrees, that's a totally different story. The problem there is that you've literally broken the enzyme. It's been denatured.
Okay, let's break down exactly where those 5 marks come from. It's really specific. You get 1 easy mark just for pointing out the obvious, that the starch never got broken down in either case. Then you get 2 marks for explaining the 5 degrees situation. Not enough kinetic energy means fewer collisions. Then, the final 2 marks are for the 80-degree situation. The amylase is denatured, which means its active site has changed shape and the starch just can't fit anymore.
All right, we're on the home stretch. This last question is all about thinking like a scientist. You've done the experiment, you've got your results, so what's next? Where do you go from here?
To get the full 2 marks on this, you need to lay out a logical next step. First mark, you say that now you'd keep the temperature constant. You've already investigated temperature, so you lock that in. And the second mark? That's for picking a new variable to investigate, something like pH, or the concentration of the enzyme, and then crucially, testing a range of values for it.
Okay, so we've made it through the whole question. What's the big takeaway here? I mean, it's about way more than just amylase, right? It's about showing you know how to do science.
Think about it. This one question tested everything. Your basic biology recall, your understanding of how to design an experiment, your ability to look at data and see a story, and even your skill at planning what to do next. It just goes to show how all these skills are woven together. And it proves that it's not just about what you know, but how you communicate it in a way that lines up with that all-important mark scheme.
So, when you boil it all down, what's the #1 skill for crushing these exams? It's not just knowing the biology inside and out. It's about understanding the game. It's about knowing exactly what the person marking your paper is looking for. And there's no better way to learn that than by doing practice questions, just like this one, with the mark scheme open right next to you. I really hope this deep dive was useful. Thanks for tuning in.